Dear students, till now we have discussed about gametogenesis, that is a pre-fertilization event, spermatogenesis in males and oogenesis in females. Now before taking up the gamete transfer, let's talk about the structure of an ovum. This is the diagrammatic view of the ovum. Like any other animal cell, ovum is also surrounded by a plasma membrane. Then it is having nucleus, cytoplasm and all the other cell organelles. Now to the inside of the plasma membrane, there are granules present in the cortex region of the ovum. These granules are called cortical granules. Then next to plasma membrane on the outside is the vitelline membrane. And next to vitelline membrane, there is another membrane called zona pellucida. And in between these two membranes, there is a space which is called perivitelline space. Now you can see some of the cells are radiating around the ovum. These cells are actually the granulosa cells of the follicles. You remember ovum development occurs inside the follicles. So when the ovum is released from the ovary during ovulation, some of the granulosa cells, they remain attached to the ovum. So this layer of granulosa cells is called corona radiata. Now let's talk about the gamete transfer. Gamete transfer in case of humans is called insemination. Why insemination? Because it is the introduction of semen into the female reproductive tract. Semen is composed of seminal fluid, which is the secretion of male accessory glands, and it contains about 200 to 300 million sperms. So what is semen? Semen is seminal fluid plus sperms. So semen is introduced into the vaginal tract, and through the vagina it will the sperms in the semen, they will vigorously swim from the cervical canal into the uterus, and they will reach the respective fallopian tubes. Now only few of the sperms are succeeded in reaching the ampullary isthmic junction. Now sperms have reached, and if there is simultaneous transport of ovum from the ovary at the ampullary isthmic junction, only then fertilization occurs. If both ovum and sperm are not reaching at the ampullary isthmic junction, then fertilization will not occur. That's why all copulations does not lead to fertilization and pregnancy. So once the fertilization has occurred, then what happens? Zygote formation occurs, and zygote will divide and redivide, and it will become two-celled, four-celled, and start moving towards the uterus. Now let us discuss about the fertilization event in a bit detail. You see here, this is ovum with its zona pellucida, vitelline membrane, and in between is the perivitelline space, plasma membrane, cortical granules. This is whole ovum or egg, and this is the cytoplasm. Now what happens? Lot many sperms they come in contact with the ovum, but only one of them succeeds in fusing with the ovum. Why? Let's see why this happens. Why only one sperm is able to fertilize the ovum? As we have discussed the structure of sperm, we have discussed that head of the sperm is having a cap-like structure called acrosome, and that acrosome produces hydrolytic enzymes. Now these hydrolytic enzymes, they help in dissolving the zona pellucida. Now you see here, this has crossed the zona pellucida layer. Then it moves further, and hydrolytic enzymes of the acrosome will further dissolve the plasma membrane. Plasma membrane of the sperm and the ovum plasma membrane, they fuse, and the sperm nucleus is introduced into the cytoplasm of the ovum. Once a sperm nucleus has been introduced, what happens? The cortical granules, they fuse with the plasma membrane, and these granules, they block from all sides, these granules, they will fuse with the plasma membrane, and these granules will block the entry of further sperms into the ovum. So no more sperm can enter once a single sperm has able to enter the ovum. Now let's discuss about the fertilization and implantation. Now this is the fallopian tube and this is the uterine cavity. This is ovary with its ovarian events occurring inside. Once the ovum is released, actually this is the secondary oocyte. Now once the ovum or secondary oocyte is released, and if it happens to reach at the ampullary isthmic junction and happens to meet the sperm, then zygote formation will occur. Now this zygote will divide and redivide. It will become 2-celled, 4-celled, then 8 to 16-celled, and each cell of these embryos are called blastomeres. And the type of division here is called cleavage, because the overall size, the size which was of the zygote, the overall size will remain the same and small cells are formed in between. Now morula is the 8 to 16 celled blastomere stage. Now 16 celled will later on becomes 32 celled, and simultaneously there is movement of this embryo. Now when it is 32 celled embryo, it gets implanted in the endometrium of the uterus. Once it gets fixed, now this 32 celled embryo is called blastocyst. Its structure we will discuss later, but before this let's discuss about the implantation process. Now this 32 celled blastocyst has fixed itself in the endometrium of the uterus. Now the cells of the endometrium, they will divide and redivide and surround this blastocyst such that blastocyst gets embedded in the endometrium wall. And this embedding of blastocyst into the endometrium wall is called implantation. Now what happens? These 32 cells, they rearrange themselves. How? This is a blastocyst with rearrangement of the blastomeres. Inner group of cells is called inner cell mass, and there is a single layer of cells on the outside, and this is called trophoblast. It is the inner cell mass from which the embryo will develop. And trophoblast will help in the formation of placenta along with the maternal uterus tissue. Now what happens? Trophoblast will have finger-like projections which are called chorionic villi. And these finger-like projections called chorionic villi, they will interdigitate with the endometrium wall, so such that placenta is formed. And placenta is a structural and functional unit through which there is exchange of material between the fetus and the mother. And embryo is connected to the placenta through the umbilical cord. So all the exchange of material will be occurring through the umbilical cord. Through this umbilical cord, through the placenta, embryo will receive oxygen and nutrients. And in the embryo, carbon dioxide and other waste so formed will be passed on to the maternal blood through this placenta. Now placenta is a temporary endocrine gland which is formed during pregnancy. It's an endocrine gland and it produces hormones, and these hormones, they are important for the normal embryonic growth. So let's discuss what are these hormones produced by placenta. These are human chorionic gonadotropin hormone, human placental lactogen, estrogen and progestogen. All these hormones are important for the maturation and development of the embryo. Now with time, what happens? 32 celled embryo will further divide and redivide, and after 1 month in that embryo heart formation will start. And actually what happens, their inner cell mass is forming the embryo, and inner cell mass is further differentiated into three wall layers: ectoderm, endoderm, and in between these is mesoderm. So this inner cell mass, trophoblast is involved in placenta formation, and inner cell mass, it further differentiates into three layers: ectoderm, mesoderm, and endoderm. And all these three wall layers will result in the formation of different organs of the body of the embryo. So after 1 month, heart formation occurs, and by the end of 2 months, limbs and digits appear in the embryo, and by the end of 12 weeks, major organ systems develop. By the end of 24 weeks, body hair develop, eyelids separate, and eyelashes are formed. Now after a period of 9 months, now the fetus is ready for the birth, and that childbirth is called parturition. Parturition is an neuroendocrine mechanism. It is a neuroendocrine mechanism because neural system as well as endocrine system involved in the childbirth. First signals for the parturition originate from the fully developed fetus, and these fully developed fetus as well as placenta. So first signals for parturition are originating from the fully developed fetus and the placenta, and these signals are called fetal ejection reflex. These signals will in turn initiate the maternal pituitary to produce a hormone which is called oxytocin. This oxytocin causes vigorous contractions in the wall of the uterus. This in turn will stimulate maternal pituitary to further secrete more oxytocin. This stimulatory reflex between the uterine contraction and oxytocin continues, resulting in stronger and stronger contractions. This leads to the expulsion of the baby out of the uterus through birth canal, and this is called parturition. Soon after the infant is delivered, the placenta is also expelled out of the uterus. By the end of the pregnancy, mammary glands also differentiate. The milk produced during the initial stages is called colostrum, and it is rich in antibodies, which is very good for providing immunity to the newborn baby. So we have discussed all the phases of reproductive events in humans. So let's see how much you retain. So exercise your brain. In the description box below the video are some questions. Try finding answers to those questions, and let's see how much you retain. Good luck to you. Have a good day.